An Introduction to Cancer Biology. In this presentation, we will discuss selected characteristics common to many cancers. Abnormal signal transduction resulting in uncontrolled cell proliferation. Loss of apoptosis or programmed cell death. Tissue invasion and metastasis, permitting spread of the cancer. And angiogenesis, leading to enhanced blood supply of tumors. Normal cells require signals, usually delivered by ligands, to stimulate their growth and to tell them when to stop growing. These signals, or ligands, can be in the form of growth factors and inhibitors, extracellular matrix components, or cell adhesion molecules. These signals are transmitted into the cell through proteins found on the surface of cells called receptors. Each ligand binds to its own specific receptor. Receptors often consist of three domains: an extracellular ligand binding domain, a transmembrane domain, and an intracellular domain. Binding of a ligand to the extracellular domain activates the receptor tyrosine kinase, which activates other proteins by phosphorylation of, adding a phosphate to, the amino acid tyrosine on a protein inside the cell. When a ligand binds to the receptor, a signal goes to the intracellular domain, activating the associated enzyme and initiating a cascade of signals to the nucleus that tell the cell to grow and divide or to stop growing. Malignant cells generate many of their own growth signals, which allows them to divide with reduced external growth stimulation. Some cells are able to produce their own growth factors and stimulate their own growth. This is called autocrine stimulation. For example, glioblastomas express platelet-derived growth factor, or PDGF. Sarcomas express tumor growth factor alpha, or TGF-alpha, as well as epidermal growth factor receptor, or EGFR. In normal cells, the production of cell surface receptors is limited by cellular restraints on gene expression and protein translation. In tumor cells, however, mutations in the genes encoding for the receptors disrupts this finely tuned regulation, and too many copies of the gene are produced, a phenomenon called gene amplification. This, in turn, leads to excessive transcription and production of receptors. The end result is a higher than normal number of copies known as overexpression of the receptor on the tumor cell surface. This gives the tumor cell increased potential to be triggered into a growth phase by the binding of ligands to the excess receptors. The more receptors expressed, the more binding sites are available for the ligands. Gross overexpression of growth factor receptors can result in ligand-independent signaling, where receptors are active in the absence of stimulating molecules. Structural changes to a receptor can also lead to ligand-independent activation. For example, truncated versions of the epidermal growth factor receptor, where much of the intracellular domain is missing, are constitutively active. EGF receptor, also known as HER1 or ERB1, is a member of a subfamily of type 1 receptor tyrosine kinases. These receptors are found primarily in the membranes of normal epithelial cells from skin, breast, colon, and lung, amongst others. EGF receptor and its ligands play an essential role in the regulation of cell proliferation, differentiation, and survival. EGFR is overexpressed in tumors arising from the colon, rectum, and head and neck, to name a few. When a specific ligand binds to its receptor, this leads to changes in the receptor that transmit a specific signal into the cell. For example, the receptor tyrosine kinase is activated and initiates a signaling pathway specific to that receptor. This phenomenon is called signal transduction. Activation of a signal transduction pathway creates a complex chain of events in the cytoplasm or fluid intracellular space that eventually leads into the cell nucleus, where the transcription of genes regulating cell cycle progression are stimulated, resulting in cell proliferation. One of the major cascades implicated in cancers is the Ras-Raf mitogen activated protein, or MAP, kinase pathway. Another interesting pathway is the phosphosinositide 3 kinase, or PI3K AKT mammalian target of rapamycin, or mTOR pathway. These pathways are linked to each other and other signal transduction pathways in the cell. Deregulation or loss of normal controls in any of these pathways is thought to be present in all human tumors. Once the signal reaches the nucleus, transcription factors are activated. These factors transcribe the genes that are translated into the proteins, such as growth factors, that are necessary to allow the cell to continue to proliferate. The end result of any growth factor receptor signal transduction pathway is tumor DNA replication and cell division. One tumor cell becomes two. If mitosis continues, 2 tumor cells become 4, with exponential growth potential. On the tissue level, this leads to increased tumor growth and increased tumor size. Many of the components of these signal transduction pathways are potential targets of anti-cancer therapies. Therapies can target the ligands, receptors, intracellular second messengers, and nuclear transcription factors responsible for tumor growth. Ligands can be neutralized before they bind to the receptors. An example of this is bevacizumab or Avastin, which is a humanized monoclonal antibody that targets circulating vascular endothelial growth factor or VEGF. Platelet-derived growth factor or PDGF and fibroblast growth factor or FGF are other examples of ligands that can be targeted for different cells in the body. The receptors on the surface of normal and tumor cells can be inhibited directly. Cetuximab or Erbitux is an example of this. It is a chimeric antibody that binds directly to the epidermal growth factor receptor and competitively inhibits the binding of EGF and other ligands, such as TGF-alpha. Another way to block the receptor's function is through small molecule inhibitors of receptor phosphorylation associated with them. For example, EGF receptors have a tyrosine kinase that can be blocked by the molecules gefitinib or Iressa and erlotinib or Tarceva. Apoptosis, or programmed cell death, is one of the mechanisms by which organisms limit the growth and replication of cells. If apoptosis did not occur, there would be no way to control cell growth and tissue homeostasis would be lost. In fact, this is one of the key mechanisms behind cancer. The genetic alterations in the cancer cell not only lead to increased cellular proliferation and growth, they also lead to loss of apoptosis. Along with too much cell growth, there is too little cell death in malignant tissue. Apoptosis occurs in normal cells to allow for removal of damaged cells, maintain a constant number of cells in regenerating tissues, and is an important part of embryogenesis. In an average human adult, 50 to 70 billion cells undergo apoptosis per day. Apoptosis is characterized by changes such as cell shrinkage, mitochondrial cytochrome C release, fragmentation of cell DNA into multiples of 180 base pairs, and the ultimate breakage of cells into small apoptotic bodies, which will be cleared through phagocytosis. Phagocytosis is a process where cells take in cell fragments or microorganisms in membrane-bound vesicles. The vesicles fuse with lysosome-containing proteases, and the engulfed material is processed for recycling. There are 2 pathways that can activate apoptosis. The first is the death receptor, or extrinsic pathway. It is triggered by activation of members of the tumor necrosis factor receptor superfamily. The second means of initiating apoptosis is through the mitochondrial or intrinsic pathway. This is set in motion by DNA damage. Both pathways ultimately stimulate a set of enzymes called caspases. The caspases interact with inhibitors of apoptosis proteins, or IAP, and the BCL2 family of proteins, which individually have either pro- and anti-apoptotic properties. In some malignant cells, there is resistance to apoptosis due to overexpression of anti-apoptotic proteins. For example, survivin is an IAP that is found in many cancers and predicts for poor outcomes, and BCL2 is overexpressed in B-cell lymphomas as a result of the translocation of its gene. Conversely, deactivating mutations of a pro-apoptotic molecule like BAX is seen in some gastrointestinal tumors and leukemias. Anti-cancer agents have been developed targeting anti-apoptotic molecules. For instance, short segments of DNA complementary to the RNA of BCL2 or antisense oligonucleotides have been designed to reduce the translation of this anti-apoptosis protein. Activation of transcription factors can lead to apoptotic resistance. This occurs, for example, when members of the nuclear factor kappa B or NF-kappa B family of transcription factors are overexpressed in certain tumors, which lead to increased transcription of anti-apoptotic members of the IAP and BCL2 families. The ubiquitin proteasome pathway regulates the expression of transcription factors and other cell cycle proteins. Certain molecules can suppress or reduce NF-kappa B and AP1 activation and inhibit tumor promotion. Bortezomib, or Velcade, is a proteasome inhibitor that has shown promising results in multiple myeloma. It inhibits the proteasome, which leads to increased levels of the NF-kappa B inhibitor and therefore less anti-apoptotic proteins. Normal cells grow in a controlled manner to form tissues that form organs with specific functions. Malignant cells are defined by their ability to invade adjacent normal structures and be disseminated or metastasize. Malignant tumors can metastasize at any point. They do so by having cells break off from the main tumor, enter the bloodstream and/or lymphatic channels, and travel to other parts of the body to initiate a new tumor. Their ability to invade eventually affects the function of the normal tissue into which they are growing. Metastases is a multifactorial process involving complex interactions between tumor cells, vessels of the lymph and vascular systems, the basement membrane surrounding those vessels, and the extracellular matrix, which consists mainly of collagen. Let's take a closer look at how the EGFR pathway activates and modulates metastases. When the appropriate signals enter the cell, a complex chain of events within the cytoplasm is set in motion. These events eventually lead into the cell nucleus, where the transcription of genes regulating cell cycle progression and cell growth are stimulated. One protein produced through this cell activation process is the enzyme matrix metalloproteinase, or MMP. When a tumor cell metastasizes, it breaks off from the main tumor and enters the extracellular space. Tumor cells secrete MMP, which degrades the collagenous extracellular matrix, or ECM, breaking through the basement membrane that surround the tumor and allowing the tumor cell to migrate toward the blood or lymph vessels. When the MMPs reach the vessel, they break down the basement membrane surrounding the vessel through enzymatic action, opening access to the epithelial cells lining the vessel. Tumor cells can then migrate into the blood and lymph by entering through the tight junctions of the epithelial cells. The tumor cells are then transported through the blood and lymph to other tissues. It is known that metastatic tumor cells tend to target some organs more than others. As the tumor grows, it will eventually reach a size where it will need to have additional vasculature to sustain continued growth. To achieve this, the tumor cells excrete certain proteins to stimulate blood vessel growth into and around the tumor, a process called angiogenesis. One of the major pathways involved in angiogenesis involves vascular endothelial growth factor, or VEGF, and its family of receptors. There are 7 subtypes of VEGF and 3 receptors that bind differently. VEGF affects the endothelial cells that line the blood vessels in a number of ways. It can cause them to proliferate by activating the extracellular kinases and MAP kinase signal transduction pathways. It can induce proteins that break down the basement membrane to allow endothelial cells to migrate and invade. These proteins include matrix metalloproteinases, or MMPs, urokinase-type plasminogen activator, or UPA, and its receptor UPAR, as well as the tissue-type plasminogen activator. It makes vessels more permeable, allowing molecules and fluids to leak out. When MMP is secreted into the extracellular space, it degrades the extracellular matrix to allow proangiogenic factors to reach the vasculature. With the extracellular matrix degraded, proangiogenic factors, including VEGF, can reach receptors on the endothelial cells of blood vessels surrounding the tumor, thus stimulating the angiogenic signal in the vessel. VEGF also helps the new endothelial cells survive by upregulating inhibitors of apoptosis. VEGF also activates the endothelial cells to express the proteins necessary to allow the new blood vessels to form. The end result is the growth of new blood vessels into the tumor. With the growth of new vessels into the tumor, additional nourishment can be delivered to the tumor. New blood vessels in the tumor facilitate further tumor growth. Strategies targeting VEGF and its receptors have been used successfully in clinical practice. Bevacizumab, or Avastin, is an antibody that binds VEGF and prevents its binding to its receptor. Another therapy is sunitinib, or Sutent, which is a small molecule inhibitor with high binding affinity for VEGF and PDGF receptors. The receptors on the surface of normal and tumor cells can be inhibited directly. Cetuximab, or Erbitux, is an antibody that binds directly to the epidermal growth factor receptor on the normal and tumor cells and competitively inhibits the binding of EGF and other ligands, such as TGF-alpha. As we have seen in this presentation, tumorigenesis is a complex, multi-step process that results from genetic changes and causes malignant transformation of normal cells. A summary of the characteristics common to many cancers are: abnormal signal transduction resulting in uncontrolled cell proliferation, loss of apoptosis or programmed cell death, tissue invasion and metastasis permitting spread of the cancer, and angiogenesis, leading to enhanced blood supply of tumors. Many of the components of these mechanisms are potential targets of anti-cancer therapies.